So welcome back, everyone. Welcome to all the new attendees. Today it's my pleasure to talk to Maude Beaudoin. She earned a PhD in 2015 working on Parkinson's disease, and she later worked on Huntington's disease in Cambridge. She is now based in Lyon, France, working on Coma, obviously, and she'll talk to us today about what Coma really is, what we know so far, and what she's been working on. So if it's your first time attending, just letting you know that you can post your questions in the chat while she's talking, and we'll just read it at the end and answer all of them. Thank you so much. Thank you, Maude. Can you just share your slide?
So do you see the screen? Yep. Okay, so thank you. So let's start about talking about coma and comatose patients. So first of all, I think the good start could be to define the word coma. What is it? It's from the ancient Greek coma, which means deep sleep with non-spontaneous awakening, no way to exit from the sleep, even if there's noise and then their pain and so on. So this is the topic of my talk. But why should we work on coma in research? It's because in the last decades, there was huge advances in the intensive care unit management, and it has led to decreased mortality of patients with huge brain lesion. But the fact is, even if we're able to decrease the mortality of some patients, there is still the fact that they survive with huge brain lesion and with a certain quality of awakening and a high risk of functional disabilities. So the research of coma aims at understanding what's happening in the brain lesion in patients. So just start about what is coma for you. So usually when we say comatose patient, you have this image, that's to say patient with no reaction at all, just like they're sleeping, they're not moving, they have closed eyes and so on. But we'll see together that it's just a picture and it's not so right. So just switch off the TV and speak about real comatose patient. So usually when we speak about coma or consciousness impairment, we describe it with two dimensions: the content of consciousnness, so that's sort of your conscience of yourself, the environment, all the memory and so on. And also the level of arousal, that's just the thing of being awake. So when you're conscious wakefulness, you have both dimensions that are at the maximum. You are fully content of consciousness and you have full level of arousal. But in our daily life, we all experience the decrease of these two dimensions. For example, when we are sleeping. When we're sleeping, we have a decrease of our content of consciousness and also a decrease of the level of arousal. And we can also experience mismatch between those two dimensions. For example, during the rapid eye movement sleep, in this case, you have a decrease of arousal because you're sleeping. But you have a small increase of your content of consciousness because you're dreaming, so it's something is happening. But in any case, even if you're on the deepest sleep possible, it's always possible to wake you up. So let's see if we can decrease again the content of consciousness and arousal, and in fact it is, by general anesthesia. So we can use drugs to decrease more your content of consciousness and your level of arousal, and it's not possible to spontaneously being awake. And around that, there is a huge part of pathological conditions which are called disorders of consciousness. Among those disorders of consciousness, first of all, there is what we call coma. So it's almost a null content of consciousness and arousal. So this is typically what we have seen in TV shows. So someone's just like sleeping and eyes closed and so on. But it's a very transient condition. And in fact, in intensive care unit, patients evolve in vegetative states. So what is the vegetative state? It is a state when there is an increase of the level of arousal, but it's almost the only things happening. So in other words, it means that patients are able to open their eyes spontaneously and close their eyes. And they can open it and close it depending on the day or the night, but not necessarily. It could be completely chaotic. But this is what you can say. You can see if you go in intensive care unit patient with eyes open and just, that's the only thing almost they're doing. The second state that we can describe is the minimally conscious state. So this is a state when there is a small increase of the content of consciousness. But the problem is, can we know exactly what is the content of consciousness of those patients? So basically, just assume that the consciousness is just like an iceberg. So there's a lot of processes happening, but we cannot see them. For example, you're conscious, you have perception, emotions, you use language, you have memories, and you have executive functions. So your whole experience is that. But how can I know that you're conscious? I can know that because we can use communications. So you're all able to say, yes, I'm conscious and I'm sitting in a chair in front of my laptop and so on, because we're able to talk together. But when there's no communication at all, and that's the case for patients, how can we assess their level of consciousness? We can use cues of cortical functions. And classically, this is what we do in intensive care unit, for example. So this is used in all clinical scales to evaluate patients. So what is a cue of cortical function? So for example, we can assess visual movement. So you can see here in patient, we can see if there's eyes movement. For example, looking your own eyes in a mirror, for example. So this is one of the things we can evaluate in patients. We can also see if you're able to use functionally objects. So for example, we can put a pen in patient's hand and see if he holds it correctly. I'm not talking about being able of writing. I'm just talking about being able just to hold it correctly, for example. This is a functional use. We also have patients in bed, so we can see if patients are able to hold the sheet and remove it, for example. This is also another functional object manipulation. So it's very simple, but this is the kind of cues that we're using in clinical evaluation of patients. So now that we know what is a vegetative state and minimally conscious state, the question is, why doing research on disorders of consciousness? So there's a clinical point of view, which is trying to refine the diagnosis between those two states. So you can easily imagine that it's very difficult just to see if a patient is in a vegetative state or minimally conscious state. So one way could be just to better the diagnosis to see if a patient is in one state or the other one. Keep in mind that in the minimally conscious state, you have a content of consciousness. So it's really important to know if the patient is somewhere conscious or not. The other thing is we can use that to identify biomarkers of conscious. And it could be used as the prognosis tool. So we can use that to see if we can predict if a patient who has early biomarkers of conscious will evolve and what will be the outcome of the patients. It's important because, again, just keep in mind that we're working on comatose patients who are in intensive care unit, and you have the family, the relatives just asking, is he okay? What will be the outcome? Will he will be awake and able of speaking and so on. So we have a lot of questions, and it could be very, it's important to have answers for them. So it could be two aims of clinical point of view. And in the fundamental point of view, just we're talking about brain cells and we're talking about their ability just to produce thoughts. So I think this is the very sort of magic point of view. So what's happening, we have these brains and we're just speaking about consciousness. But let's see that in a clinical point of view. So just historically, in fact, some say that coma is sort of pathological sleep. And in sleep, we're able to wake up, but it's not the case in coma. So perhaps the system involved in sleep is dysfunctional in coma. So the first research about that look at a brain region which is responsible of the sleep-wake cycle. So this is the brainstem that you can see here. So this is the part of the brain linking the brain and the spinal cord. And if you look at what's happening in the brainstem, you have a region named, and it has been shown area, you can have sort of comatose state. Okay, this is the coma-causing lesion, but in fact, it has been shown that it's not so simple, and it could be also related more to arousal dysfunction, but not to the content of consciousness. So we need to go further to understand what's happening. There's a huge literature about coma, so I will just focus on one way to study that, which is the cortical connectivity. So one way to research on coma, to do research on coma, is to look at the connectivity between cortical regions and their ability to work together. So we can see if there's efficient connectivity between brain area close, and also with long distance areas. And of course, if there's connectivity between those two hemispheres. And now one way to investigate that is to use the graph theory. So the graph theory is a way to analyze, for example, the connectivity between all those events, all these dots, and to build a network and to identify if there's some points which are very connected to everything. So that's what we call hub. This way of analysis can be applied for huge things. For example, we can use that to identify hub between people due to the social interaction, for example. But you can also use that in brain imaging. So just imagine that we're looking all the connection and the interaction between all those brain area. So this is basically what we're doing in patients, for example. And there's huge literature just has demonstrated that there's cortical hubs in a network that is named the default mode network. So basically, this is the sort of brain network that is activated when you're doing nothing. So there's cortical hubs related to disorders of consciousness in the prefrontal cortex. So this is the frontal part of the brain, and also in the parietal, which is here. And there's another hubs here in the insula. So it's more like a hidden part of the brain, in the cortical brain, but it's involved also. And here in the cingulate cortex. So all the cortical connectivity identify that hub, saying that, okay, if these regions are dysfunctional, so you can have disorders of consciousness. But we can see what's happening if we're modulating the consciousness and the arousal in a healthy brain, and to see if the prediction of those graph theories are just confirmed. So this is a study that has been realized in monkeys. So you just have monkey in functional MRI, and you have sedation of this monkey, and you can see if the graphs are the same in those different states. So you can see all the brain regions that have been identified and characterized the graph of an awake monkey. And this is for the moderate sedation. So you can see that there is a decrease of the network and a decrease again in deep sedation. So this study, in fact, there's not different graph in these states, but just the same, which is dysfunctional and smaller and smaller and smaller. So it could be difficult to use that to identify if someone is awake or not, because basically it's sort of the same network, rich or less rich. And in fact, what's happening is the thing that characterizes the state of awake or not is the fact that when you're awake, you have all those brain region, all those network and all the graphs that are just moving very fast between them. So in fact, if you're awake, it's not because you have an awake network. It's more that your brain is able to change network very quickly, and you're not able to do that when you're unconscious. So this is for the moment where we are for the network analysis. But this is only with cortical region. And you know, and we know that the brain is not only a cortex, and there's huge parts of the brain in the subcortical area. So within the brain, you have all the subcortical area that are connected to the cortex using loops. So for example, you have the cortex, which is connected to the striatum, which is connected to another part, which is named pallidum, and connected to the thalamus, for example, and go back to the cortex. So you have always those loops with the feedback to the cortex. And the thing is, when you have dysfunctional cortical network, perhaps it's because there is dysfunctional subcortical areas, and in the cortex, you just see the echo of what's happening. And there's some cues about that in the literature. So first, for example, it is known that if you have thalamic infarctus here, so here is the thalamus, and if you have an infarctus in that, just in that, you have disorders of consciousness. And if you look at the metabolism of the brain, that's to say the activity of brain region, you can see that in healthy subject, you have metabolism, so activity in the thalamus, which is decreased in a patient in a vegetative state. But on the opposite, in this more recent paper, so you have a patient in a vegetative state who have stimulation of the vagus nerve, and there was a slight improvement of his clinical state. And this improvement was linked to an increase of the metabolism in the thalamus and the striatum. So all those elements show that it's also important to look at the subcortical structures and the way that there's communication between subcortical and cortical structures. And this is exactly what we're doing in the research project I'm working on. So in Lyon, we're working on imaging the neural basis of disorders of consciousness. So we're working in patients in intensive care units. So you have patients that are admitted in the intensive care unit after traumatic brain injuries, ischemia, hemorrhage, and all patients are intubated. So keep in mind that we are very early close to the brain injury, and patients are very sensitive. Due to the clinical management at a certain time, the intensivists stop sedative drugs. So it is classic that patients in intensive care unit with brain lesion are sedative at the moment, and we stop the drugs. And if there's no sign of awakening after the cessation of sedative drugs, we do imaging, brain imaging in a PET MRI device. That's to say, we're just imaging a lot of brain parameters at the same time. And after that, we were used to do a follow-up during 1 year to see what will be the outcome of the patient, and to see if we can identify biomarkers very early here when we're doing MRI, and if we can link those biomarkers to the patient outcome 1 year after. So this is classically what we're doing, and you can see here, this is the brain of our first patient, for example. So just an example, we're recording the brain metabolism, that's to say the ability of brain region to use sugar to work, but we're also doing functional MRI to see what brain region are working all together. And we're also doing tractography. This is an anatomical analysis when we can just follow anatomically all the neurons. So this is what we're doing in theory, because you'll see that in comatose patients, it's not so simple, because honestly, this is what we have in reality when we work with comatose patients. So we have to face a lot of methodological challenge. For example, you have huge brain deformation here, for example, because of edema. You have artifacts due to ventricular drainage, for example. So it's not, there's not missing part of the brain here. It's just that there's artifact because of medical things there. And you can also have here a huge brain deformation because of decompressive craniectomy. So this is not an artifact. This is the real shape of the brain in the skull, okay? And you can also have a lot of movement here, for example. So in fact, working on comatose patient is first facing all this methodological challenge. And this is why there's not a lot of studies in intensive care unit, because it's really difficult to have all this imaging in those patients. But so that's a study which is going on. So we're just doing imaging in 90 patients. And the aim of this project is at the end to build a prognostic tool and to see if we can just identify biomarkers to see what will be the outcome of patients. And okay, obviously I'm not alone working on this project, and I think it's important to show you all the people involved. So first, of course, I'm a neurobiologist and I'm working on patients, which means that in fact I'm working with MD, PhD with patients in the intensive care unit, but also in the rehabilitation department. And in Lyon, we have all these labs and departments working with us to build that prognostic tools. And obviously research costs money, and we have money from national and local grants just to help us to do this project. So we spend a lot of time finding money, and so I think it's important to tell when we can finally have it. So thank you, everybody. And I hope you enjoyed the talk, and I would be very happy to try to respond to your questions.
Thank you, Maude. Really nice talk. I really enjoyed it. As I said, if you have any questions, put them in the chat. I'm just going to start to just give you a little bit of time to write. Just curious, are there any animal models of coma?
Yeah, so there's a lot of rodent model of coma, but in fact, the thing is, there's animal model of brain lesion. So not in coma itself, because it's assumed that there's no way to evaluate the consciousness of animals. For example, how can you say that a rodent is awake or not, is conscious or not? So scientists decided not to ask this question, and they look at brain lesions. So you have traumatic brain injury model in rodent, and you have ischemia model in monkeys, for example. So the thing is that now, with all the ethical guidelines that we have, there's a lot of things that we cannot do anymore. And I think it's good because there's a lot of studies in the 80s about that. And I think now we're trying to develop animal models that are more ethical, and it's more sedative model, for example, just injecting the anesthesia and see what's happening in an MRI, for example.
Okay. So there is a first question from Felipe saying, can you distinguish coma arising from a traumatic event from one arising from a vascular event, like another disease?
Yeah, so if I understand, if we can just see different mechanism between ischemia and traumatic and so on. That's right. That's
what it means.
Yeah. Okay. In fact, it seems that there's very different mechanism involved in that, because, for example, if you have an ischemia, because, for example, I don't know, heart cessation, you have all the brain that has been as involved in an ischemia mechanism. Whereas in trauma, you have those local phenomena with a lot of inflammation, injury, and so on. So, in fact, the thing is that there's a lot of literature just looking at ischemia and what's happening in ischemia and not in trauma. So it seems that it's not the same thing, and there's perhaps common mechanism, but there's no study comparing all of this.
So a new question, Caroline, saying, do you mean that coma is close to anesthesia?
The thing is that clinically it's close because you have the same clinical manifestation, that's to say no consciousness and no arousal. But when you look at what's happening in the brain functioning in anesthesia, there's perhaps some common pattern in the default mode network. But the thing is that we assume that anesthesia is in an healthy brain, and it's not the same thing as a traumatic brain, for example. So there's a lot of literature saying, okay, we just put propofol to people and say it's the same thing as coma. But obviously it's not the same thing, because at the moment you stop the propofol and people are just awake, which is not the same in brain lesions. So perhaps there is additional mechanism in patients.
Kagan is saying, thank you very much for this fascinating talk. To your knowledge, are there any changes in brain perfusion and or drainage in CSF?
Okay, I'm not sure to have the question, because my web connection is not so good. So I'm just trying to have the.
Are there any changes in brain perfusion and or drainage
okay.
CSF?
Okay. So yes, there is, and basically in comatose patients, there is a strong decrease of the brain perfusion, for example. So you can manage that in imaging, and you can see that there is a decrease. And the thing is, we try to know if there's decrease in some region, for example, to use it as a pattern of if there's a global decrease. So, for example, in ischemia, in a global ischemia, in anoxia, for example, you have a decrease in the whole brain, but it's not the same, for example, in trauma. So you can have increase also of the brain perfusion if there's inflammatory mechanism, for example. So again, each patient is different and each etiology is different, so you can have different mechanism in that.
Laura is saying, brilliant talk. In the comatose state, the patient is not so aware of the external stimuli or in the vegetative state. You get that. She's asking if the patient is, I think that's what she's asking, if the patient is aware of external stimuli.
Well, it depends on what we call external stimuli. For example, if the reaction of the pain is external stimuli, so you can have reaction, for example, just removing their hand if there's stimulation, nociceptive stimulation. You can also have reaction, for example, if you enter in the room, you have the patient that look at you and follow you, for example. So this is sort of reactivity of the patient. So you can have that, but the thing is, we don't know if it's sort of reflex reaction or conscious reaction or voluntary reaction. So usually in vegetative patients, they can just open their eyes and look at you, but we don't know what it means.
Okay, like if you watch very naively movies, they would just tell you like, oh, you can talk to coma patients, they can hear you and everything. Is there any data on that?
Yeah, in fact, usually when patients are just discharged from the intensive care unit and you talk to them weeks after, they have no memory of what's happening in the intensive care unit. So I don't know if they listen to us, but the thing is that they don't remember they were. So you can have also reaction of an emotional content. For example, I speak to patient, there's no reaction because I'm a stranger, but her mom or a son or some just talk to a patient and she will react and smile and so on. So perhaps it's because they're just listening a familiar stimulus, but we don't know if it's a conscious process or just a reflex because it's familiarity.
So if you have, so here's my question actually. So if you have all those areas that are interconnected, so many of them, is there actually any hope of being able to map all the biomarkers that could be used to screen for any kind of consciousness disorder?
Yeah, so thanks to all the deep learning and machine learning and so on, this is what we're trying to do. So we try to scan a lot of patients with several etiologies and so on and see if we can just have all the biomarkers and to see for have dysfunction in the thalamus. It means that whatever will happen, there will be no recovery. But the thing is that as there's a huge heterogeneity between those patients, we can just assume that we're not sure that we will be able to have all those biomarkers all around. You have so much lesion and brain information and so on that I think if we can have 1 or 2 biomarkers of area, it would be nice. But you we have patients with a lot of the brain, and the half of the brain is missing. So at this moment, what can we say if we have a biomarker saying, okay, it's something is happening and so on? We don't know exactly. So, and the other thing is that it's an anecdote, but when we use machine learning, so for example, you can have algorithm that just because in the metadata, meta description of the file, they just found that it was from an hospital. So we need to be careful about identifying all those biomarkers because I think it's not so easy, and perhaps we won't be able to identify a lot instead of comatose or not. So we are very precautious about that.
And are those pathways redundant? Because on what you've shown, seems that there seems to be a lot of interconnection. Like usually in biology, when you have so many pathways, they end up being quite redundant. So if one is damaged, just another one will pick up, and you barely see the difference. But those areas that you mentioned earlier, that was 5 or 4 of them that were really involved in consciousness, are they redundant, or if any of them is depleted, you're going to be sick?
Yes, I think these 2 things. There's redundancy because we have 2 hemispheres, so we can just hypothesis that when one hemisphere is lesion, the other one can just compensate. And in fact, there's some patient that after some weeks they will be awake, and probably because they were able to compensate. But the thing is also that in the brain, at the moment, you have all the projection that converge in one region, for example, the thalamus. So we can imagine that, for example, if there's huge lesion in the thalamus or in the reticular formation, there won't be any parallel pathway to compensate. So I think there's redundancy, but in a certain point. And if there's key region, perhaps it won't be enough. So I think the important thing is to identify those key points to see if is enough to have a recovery or not.
Okay, thank you so much. That's it for me. If you have any more questions, please put them in the chat. Please raise your hand so I can know that you're typing. Otherwise, that will be to not. Oh, there is one. Clicking in again. Saying, I agree with you that the presence of massive brain lesions make human studies difficult. What is your opinion about the swine model, a pharmacological principal lesion developed in Pennsylvania? Have you heard about it?
No, I'm not sure that I heard about this model.
So if you want to comment on that, Karine, I'm going to unmute you, and you can say if you want to develop your question.
Yep.
So Karine, you should be able to talk now if you want to develop, or you can say that you don't want to. It's fine. She's hearing us. I think she's shy. She said, no problem, we'll talk about it on Twitter. Okay, well, those who are interested, I'll share the answer to everyone. Okay. Well, thank you so much. Is there any other question? Not sure there is. Thank you so much, Maude. As usual, this talk will be put on YouTube with a little delay. Will also be available on SoundCloud. Everyone is saying thank you, and yeah, thank you so much. Bye, everyone.